DNA delivery. So in this regard, DNA electroporation appears as a breakthrough in DNA vaccine field, and I will not detail the mechanism of DNA electroporation since it has been very nicely presented by Kate Broderick. I would just like to emphasize that a dramatic increase of magnitude of induced responses was observed for DNA vaccine administered by electroporation and DNA vaccine against different viruses, HIV, HCV, HBV, influenza, in different animal models, including non-human primates. So concerning in vivo DNA electroporation for HBV infection, the first studies show in large animals such as cattle, rhesus macaque, pig, that electroporation-based delivery of DNA vaccine targeting HBV envelope protein is able to considerably enhance the magnitude and breadth of cellular immune response, and as well as humoral response by 1 or to 2 order of magnitude. However, the two essential questions, what is the benefit of electroporation for viral neutralization and what is the therapeutic efficacy of this approach, cannot be answered in this model since a pig or cat are not susceptible to HBV infection, since HBV infects only human and chimpanzee. So in this regard, a DHBV-infected duck model is quite relevant for study of the impact of electroporation on prophylactic and therapeutic DNA vaccine delivered by electroporation. So I will just briefly show you that this model is a reference for human HBV study. So this DHBV virus has, as you can see here, a very similar genome organization. And it is a reference for analysis of HBV replication, not only in vitro, in primary hepatocyte culture, but also in vivo, since a chronic infection of ducklings leads to 100% of infection. And this is the actual reference for evaluation of novel therapeutic approaches against hepatitis B and DNA vaccine, and our group have developed a DNA vaccine in this model during the last few years. So the goal of the study is to break immune tolerance, stimulate B and T C responses, and the target here is the clearance of this cccDNA, which is a template for viral replication. So we have first studied the electroporation of DNA vaccine targeting DHBV large envelope protein in naive ducks, and we have used here this plasmid encoding this large envelope protein, and the electrodes which have been designed specifically for ducks by ICOR San Diego. So this is just the protocol, so there was just only 3 DNA injection delivered for electroporation. So the first question was the magnitude of induced humoral response. And as you can see here, there is electroporation, which is shown here in plain lines. So the delivery of a very low dose here, 10 micrograms of DNA, here in red, induced high antibody response here, which was quite comparable here to that elicited by a 15-fold higher dose of DNA administered by standard injection here in dotted lines. So next, we wanted to know what is the impact of DNA electroporation on neutralizing humoral response. And so the sera from these animals were tested for neutralization in primary duck hepatocyte culture. And as you can see here, a very low amount of viral DNA delivered by electroporation induced here a high neutralization titer, whereas the same amount of DNA delivered by standard injection was not neutralizing. In addition, this response was multispecific, and I have no time to detail these results. So the essential question was, of course, the therapeutic benefit of DNA electroporation in chronic DHBV infection model. So this is just the device which we have used, and we asked what was the effect of electroporation of break of immune tolerance, viremia clearance, and clearance of this cccDNA. So this is the protocol which we have used. We have established a colony of chronically infected animals by inoculation of neonatal ducklings with DHBV. So these animals receive 9 injections of DNA, which was delivered either by conventional injection or by electroporation, and this DNA encodes a viral process, envelope protein, core, and interferon gamma. And we wait here 4 weeks after the last DNA injection and the sacrifice of these animals to see if there is a rebound of viral replication. And at the end, we have analyzed the intrahepatic viral DNA replication in necropsy liver samples. So first of all, this is the impact of this strategy on viremia. And as you can see, none of the animals, here you have individual viremia titers, none of the animals from a control group or standard DNA injected group cleared the viremia, whereas we have several animals here from electroporation treated animals which have undetectable viremia. And when one look here on the mean value, there is a significant decrease in mean viremia titers in electroporation treated group, starting here from week 30. And importantly, this effect was sustained since there was no rebound in viremia after a therapy cessation. So next, we have analyzed the liver necropsy samples at the end of this therapy. And as you can see here, there is a decrease in viral replication in this group as compared with other group, and importantly, there is a significant lower number of cccDNA, this recurrent viral DNA form, in this group, as assessed here by a qPCR. So next, research for the restoration of anti-priase antibody response in this chronic DHBV-carrier ducks. And as you can see here in green, there is a strong and sustained seroconversion to anti-priase, anti-envelope response in animals which receive DNA by electroporation, and which was here significant during the last 11 weeks. So another important issue was the recognition of DHBV B-cell epitopes by induced antibodies. And here we have tested this sera from animals treated either by electroporation or standard DNA injection using the PEP scan, which cover here overlapping peptides covering the entire envelope protein. So as you can see, the sera generated, obtained from animals which receive DNA delivered by electroporation, recognize a larger number of antigen inclusion, and in particular several neutralizing epitopes as compared from those which receive DNA administered by standard injection, and there was almost no response from in the control group. So electroporation induced a higher magnitude and breadth of humoral response. So finally, we have tested here the restoration of Th1 cytokine response in the liver of these animals. And here there is, electroporation delivery of DNA, as you can see here in green, significantly increase interferon gamma expression, which is 10 to 20-fold greater than in standard animals which receive here standard DNA injection. And finally, we have tested here, we have shown here the expression of DHBV envelope protein and CD3 plus T cell infiltration by immunochemistry. And you have here, just as an example, different profiles of infection resolution. Here, there is a typical chronic DHBV infection in one animal which is received DNA vaccine administered by standard injection with massive DHBV expression and very low CD3 infiltration. And in two following panels, there are clusters so these animals receive a DNA electroporation. There are clusters of DHBV pre-S expression strengthening cells and moderated to mark portal infiltration. And as you can see, there's only very few cells which are positive. And this is one animal which show a complete resolution of infection and very early clearance of viral DNA. And this is the last slide. And this animal completely clear the infection. So in conclusion, in naive animals, DNA electroporation dramatically enhance magnitude, breast, and neutralization potency of humoral response. And just electroporation improves dose efficacy of DNA vaccine and may be effective at clinically feasible dose. And importantly, the therapy of chronically infected animals by DNA vaccine delivered by electroporation significantly give a significantly lower viremia and cccDNA level and a break immune tolerance, and also lead to the recognition of broader range of B-cell epitopes, higher level of intrahepatic interferon gamma RNA, is safe, and I have enough time to detail these results, and therefore appears to have a significant benefit for chronic hepatitis B therapy. And I just want to mention a first clinical trial of DNA vaccine delivered by electroporation against chronic hepatitis B, which has been held in China and has been recently published. And in this study, there is a combination of DNA vaccine targeting envelope and with IL-2 and interferon gamma encoding plasmid. And these results are very promising, and you can find more results on clinicaltrial.gov site. So the last but not the least, I just want to acknowledge our team and ICOR San Diego, which help us with electroporation. Thank you for your attention.
Merci bien. Are there any questions? In the back. Yes.
Hi.
Surprised by the very frequency and short time period of the vaccination. Do you really need to give the vaccine every 4 days for 3 weeks?
No, we have not given the vaccine. No, this was weeks, you know. We give the vaccine always each 3 weeks.
Oh, it looked like days.
Yeah. So.
And a follow-up, do you think you need to have sustained immunization, or will you be able to stop immunization at some point and have a sustained effect?
Well, we just stop, in terms of therapy, we have just stopped the immunization after 9 DNA injection, and we have followed these animals for 1 month, 4 weeks, and there was no rebound of replication in these animals which appear as sustained responders.